Welcome, everyone, to today's webinar organized by Cell Press on challenges and opportunities for active matter in biology. My name is Shreya Mittal, and I'm a scientific editor at Newton, the Cell Press journal dedicated to physics research. I am co-hosting this event with my colleague, Deborah Taylor, who is a senior deputy editor at Current Biology, and we will co-moderate the panel discussion. My colleague Andrew Bissett, who is the editor-in-chief of Cell Reports Physical Science, is unfortunately not able to join us today. Next slide, please. All these three journals, Newton, Current Biology, and Cell Reports Physical Science, as well as iScience, publish research related to the topic of today's event. We also encourage all of you to consider Cell Press multi-journal submission that helps you find the right fit for your research paper quickly and easily by considering it simultaneously for multiple Cell Press journals of your choice, including Newton, Current Biology, Cell Reports Physical Science, and iScience. Next slide, please. With biologists using physical principles to tackle interesting questions and biological phenomena providing useful systems for physicists, we are excited to be discussing today the many connections across these various disciplines to address challenging and open questions in the field of active matter and opportunities for the future. Today, we are honored to have three distinguished speakers for this panel. Lisa Manning is the William R. Keenan Jr. Professor of Physics at the Bio-Inspired Institute at Syracuse University in New York. Professor Raman Golestanian heads the Department of Living Matter Physics and is the director of the Max Planck Institute for Dynamics and Self-Organization in Göttingen. And finally, Professor Guillaume Salbro leads the Theoretical Physics of Biological Development Laboratory at the University of Geneva in Switzerland. Following presentations from our panelists, we will have a Q&A session, which we hope will provide a platform for engaging discussions. So we look forward to seeing questions submitted from members of the audience, from all of you. Before we get started with the talks, we have a few practical points. To submit your question, please use the Q&A box on the right of your screen. You can enter a question at any time and upvote any questions you would like to be answered by clicking on the thumbs up under the question. We will try our best to pass on as many of your questions to the speakers today as we can during the Q&A. It would be helpful if you could include the name of the speaker to whom you would like to direct your question as well. And finally, at the top right of the screen is an option to turn on closed captioning. So you can click on the CC button and select your preferred language. And now we will move to the presentations by the speakers. And as a reminder to all of our panelists, please adhere to your allotted time of 12 to 15 minutes. Kicking it off with our very first speaker, Lisa Manning. Thank you very much for joining us today.
Thank you. Thanks so much to the organizers for the invitation. I'm going to attempt to share screen again, and we'll see if I'm successful this time. I think that you can see my screen now. Is that right?
That's right.
Oh, perfect. Okay, so hello, everyone. I'm also delighted to be here with some outstanding colleagues. And so since I am first up to talk about active matter in biology, the idea that I wanted to discuss in this introductory set of slides is what do we gain or what do we learn by thinking about biological systems as active matter or active materials? And I have some videos up here and I will explain what they mean or why I think they're relevant as I go through the rest of this little discussion. So before I start talking, because I'm going to show a little bit of my own work at the end, I always like to highlight the people who did the work. And so I just want to say thanks to a bunch of folks. Science is a collective enterprise, and none of what I'm showing you at the end would be possible without these fantastic people. And I'll try to highlight who did what when I get there. Okay, but the first thing I thought maybe it would be useful to do is from my own perspective, how do I think about what active matter means and maybe set up the stage for why it's relevant for biology. So when I think about materials or matter, or the study of matter, the types of questions that scientists who work on materials think about is, what are the material properties? So by material properties, I mean things like shear modulus or elasticity, viscosity, so how easy it is to pour or flow a material. Things like thermal conductivity or electrical conductivity, whether they're optically transparent. So those type of things. And the emergent behaviors, so by that I mean, like, what does the group of things do together? Does it have some properties that I can measure or think about that only happen at this larger scale? When it's composed of many smaller scale constitutive parts. So the deal is, you have information about like the atoms or the particles that make up the material, and then you try to predict those material properties and emergent behaviors. That's typically how we do it. And now we're adding the idea of activity. And so you're asking, what happens when energy is injected or released at the smallest scale of the constitutive parts and is not given by a thermal temperature? So, typically in the past, when you thought about materials, they had a thermal temperature, and that led them to jostle around a bit, but it was pretty boring. And you could use these tools of equilibrium physics to describe them. But when you relax that constraint, you get a whole zoo of like amazing and exciting and interesting behavior. And so that's what I mean when I talk about active. Okay. But the other point that I wanted to make is the emergent behavior of dead, non-alive, non-active matter is already really hard. These are difficult problems. So I love this cartoon by XKCD creator, especially when I go to my physics department and talk to my physics colleagues. And I say, look, if you work on sand, it's way harder than any of these other things. I don't know if that's actually true. But I will say why it is difficult. So if you want to ask questions like, is a collection of sand particles, of sand grains, hard? Is it soft? Does it flow? Is it a solid? It's difficult to predict that behavior, even in an idealized setting in the lab. And so here's a picture of some elastic spheres. And what you're seeing here is force chains in the elastic spheres that are shown by photoelastic disks. And the point is that the collective behavior of a pile of sand is complex, it's self-organized, and you really can't easily predict it just by knowing that each sphere is perfectly elastic and there's nothing interesting otherwise about it. Okay. And so the difficulty of the problem is on how the interactions between the simple parts generate large scale behavior. And that's what I mean because I've used the word emergent without defining it. So to me, emergent behaviors are very interesting. And it's basically when you get some new or unexpected type of behavior at the large scale that was difficult to predict based on the individual units. Okay. And so then, of course, this was, non-alive matter. But as soon as you add activity, especially to dense matter, which is the focus of what I'm going to be talking about mostly today, makes it even more complex. So it looks like, to me, in this beautiful experiment out of Olivier Dauchot's lab, that this thing is shearing. It's like you're shearing it from the boundary, but that's actually not what's happening. Each one of those little individual disks in this packing is self-propelled. It's injecting activity at the smallest scales, and somehow it leads to this emergent behavior that kind of looks like a crystal that's shearing itself. And, if you go to even higher densities, these are just some simulations done a long time ago now by Silke Hienkes and collaborators. And the point that I'm showing here is that if you go from even in pretty dense, so I would say the thing on the left is fairly dense, if you make it even more dense, then the behavior changes dramatically. And so there's all of these interesting emergent behaviors. Because I think we'll hear about it later, I wanted to show one phase diagram in case it was interesting to folks. So on this axis is the magnitude of how fast the particles are moving. And on this axis is the density. So those were kind of the two things that I was talking about. And the point that I wanted to make is there's this really cool thing that happens emergently called motility-induced phase separation. And that's the bright red thing over there. And then if you continue to go to higher densities, you get what's called glassy or sort of solid-like behavior. And there's sort of a region in between. And I wanted to say that exciting emergent behaviors that physicists can make predictions about happen here and over here in these two different regimes. And for the rest of today, I'll be mostly focused on this blue region over here. Okay, so I talked a little bit about the physics of active matter, but the focus of today is why do we care if we're interested in biological problems? And so I'm going to talk about an answer from my perspective. There's lots of answers, but one of the questions that I would like to know the answer to is how do you I'm interested in morphogenesis. And the way I would frame morphogenesis in simple terms is how do you turn a blob of material into something that's the shape of a fish? So many of you may be familiar with this movie. This is the yolk of the zebrafish, and these are the cells that are going to become the animal at the end of the day. And there's lots and lots of divisions, and then there's a certain point at which the cells start to flow, this beautiful flow over the surface of the yolk, and you can start to see the development of the body axis. And this is the head up here, and this is the notochord and the vertebra and the tail, and you end up with a fish. And so if you're trying to ask, well, from a physicsy perspective, how might I be able to drive this amazing self-organized orchestrated thing? And there is something that's an analogy, and so this analogy was suggested by my colleague Ale Mongera, and I love it, which is that you can also take a glass blob, and if you are a skilled glass blower, you can create something that's the shape of a fish too. And what's that paradigm? How does that work? Well, I would say there's two key things that you need to turn something into the shape of a fish if you're blowing glass. One is you need to apply localized forces, and glass blowers use a tool called a mandrel rod. They also use the surface of a heat-resistant table in order to apply forces in exactly the right places and times to pull out, for example, or to push in. And the other key thing, the reason people use glass, is because you also need to alter the local material properties of the system in order to get large-scale morphological changes. If you had a very stiff glass, even if you applied a lot of force on it, you would never get deformation into the shape of a fish. And so the fact that by changing the temperature for a glass, you can change the system to go from a solid and then into a fluid by melting the glass and then back into a solid really allows you this elaboration of shapes. And so one of the cool things that I would say has happened over the past 5 or so years is that tissues do apparently change their material properties, how fluid-like they are, during development. So this is a paper by Mongera out of Oche Campas's lab, and then this really lovely paper, Heisenberg, Caneso, and collaborators that show, and these both happen to be zebrafish examples, keeping with the theme, but they show that there's a really obvious transition from a solid to a fluid that allows this to happen. And in my research group over the past 10 years or so, more than that actually, we've learned and tried to characterize the multiple physical mechanisms that can control the material properties of tissues. So this is a phase diagram, and all the stuff that's in gray is solid-like, and then these axes say, well, how can I change something? One, we've already discussed, is change the density of the material. Another that we've started talking about, and is the theme for today, is that you can increase the activity, like tension fluctuations or cell motility, or things that the cells are actively doing in order to change the fluctuations in the system. That can also fluidize. And then there's a third thing, which has been the focus of my research group for a long time now, which is this understanding geometric incompatibilities that happen in systems that are confluent or in fiber networks. And there can be interpolations between these different processes. And I wrote a review article about some of the mechanisms here in case that's useful for folks. But I wanted to take the last, I think, a couple minutes that I have to say what I think, okay, that's sort of, I would say, where we are. We understand at a basic level how active forces and interactions between constituent parts of material generate these like starting point for thinking about morphogenesis or large scale deformation of tissues. But I think the most exciting thing going forward is how do you exert this spatio-temporal control on active forces so it flows right where you want it to? You first have to turn it into a fluid maybe to get it to flow, but then you have to apply forces to stretch it. So how do you turn a glass into its own mandrel rod is like the way of phrasing it in my mind. And I was just going to highlight a couple of things that we're thinking about, but I think there's a lot of very cool stuff being done by other folks too in this space. So one of the things that we've been thinking about is the fundamentals of how to control flow in dense granular materials. And another thing is how to use active tensions in vertex models for confluent biological tissues. So one of the things that we've been thinking about is here's like a packing of like those elastic spheres up in this corner again. And now imagine that each sphere can have a self-propulsion attached to it. So there's a red arrow attached to every particle. And we're really trying to think carefully about how in this really dense system I'm going to flow, because normally such systems would be solid. And the way they flow is actually lots of work by scientists and material scientists has shown through localized deformation. So here's a picture of like a little localized deformation in a packing of active rods that allows a rearrangement. And then there's like another place where it rearranges, etc. And so it's an extremely fun but challenging problem because it means you have to understand what's the field of underlying defects in a disordered granular solid and how those interact with an applied force field to generate flow. And so we've been doing a lot of work in that space over the past couple of years. And the bottom line is by controlling the active forces, we think we can change the properties of these defects and we can actually force the system to flow where we want it to flow.
Lisa, you're at almost 12 minutes, just a time check.
Perfect. So this is, I think, my last slide. So the other thing that I'm very excited about right now is I'm thinking about how you might really concretely program those types of active forces. And in vertex models for biological tissues, one of the parameters that you think cells might have control over is active tension. So this is a vertex model, and each one of these interfaces has a tension on it. And if you wanted to think about it, you could think about that tension on each interface as possibly a tunable degree of freedom that the cell could control via, biochemical signaling feedback. And so what others have been thinking about is this new emerging idea of physical learning, which is thinking about, AI works and machine learning often works by deep neural networks with weights on each individual branch of the neural network. But actually, if you think of these active tensions in analogy to weights on a neural network, there's a whole bunch of them. And if you can tune them via feedback loops, you might be able to program this as a material, almost like you would program a neural network during a training run to make it execute a task. And so we've been thinking very concretely about this. And I'm just showing a simulation. I'm not going to talk about any of the details, but basically using ideas from backpropagation in AI and deep neural networks in order to figure out how tissue should ideally change its tensions to execute specific tasks. Okay, so I will finish there. Thank you.
Thank you, Lisa. Now we'll move on to our second speaker, Ramin Golestanian.
Okay, so let me share my screen. And presentation mode. Okay, so I'd like to talk to you about living matter as active matter. So that's basically summarizing the point of view that we just heard about from Lisa. I can maybe pose a very specific question so that the point of view of physics here is clear. So suppose we look at a living cell, that's what we have here on the right. And then suppose we have a friend who is a very good chemist, and we ask them to synthesize or collect or purify every single molecule that you can find in this living cell and put them all in a sack. That would be what you have on the left, but not in the right order necessarily, and supply the energy which is provided usually in the living cell, and then ask the question, okay, now that you have put the chemistry to one side, what would be the sort of dynamical description of this complex system such that these molecules will evolve and they find themselves in a situation that they can create this spatio-temporal organization, capable of all the things that a living cell is capable of. So that's really the question. If you want, in my view, this is what active matter is about as a field. So naturally, I would like to highlight that I think there is always going to be new physics to be discovered, because we are talking about the realm of non-equilibrium phenomena. So material properties, as Lisa mentioned, collective or emergent properties of materials when they are not active are already quite complex. And then when you make them active, you add another dimension and you lose access to many sort of solid parts of the laws of physics that you normally use the help of to describe the properties of equilibrium systems. For example, things like fluctuation dissipation theorem, or the laws of forgetting about the memory when you have equilibrium conditions, and many other things. So that would be the ideal situation where we would use living systems to try and understand and explore new physics. But along the way, when we basically learn how to do that, we will naturally also come up with blueprints of good ideas or novel ideas to make things that behave like living systems. And then effectively, with the knowledge that we have, because we have developed all those ideas from the bottom up, we can essentially make very complex systems about which we know basically everything. So one example in the context of active matter is catalytically active particles or chemically active matter. If you think about this construction of a Janus particle, which catalyzes chemical reaction in an asymmetric way, bringing in polarity and the source of energy, which will couple to the local physics to lead to motion, then essentially you can ask the question of the collective behavior of many of these particles when they are put together. And because you know everything about the mechanism and why they move and the, let's say, environment that they create around them, you can also understand how they couple to each other and how the coupling leads to emergent instabilities and properties, and also in mixed populations and so on. One thing which I would like to focus on for most of today's presentation, essentially as a development in the field of active matter, which has been very exciting in the last few years, is that of non-reciprocal active matter. So this is a situation where particles would interact with each other effectively in a non-reciprocal way. And you can very easily see why in the context of chemically active systems this can be possible, because the systems will have some locally defined way of creating non-equilibrium conditions. So what you can call the chemical activity, the flux of the reactions, so the particles coming in and going out, described by this parameter alpha, and then the response of the particle itself with gradients that are maybe generated by the particle themselves or maybe generated by other particles in the surrounding. So you can call that a mobility that's like an Onsager coefficient or a response coefficient. Now, if essentially two particles are not the same and they're both catalytically active and they have the mobility to respond to the gradients of the other ones, you can immediately see that, for example, by using the stationary solution of the diffusion equation, which looks like the gravitational or electrostatic potential decaying as 1 over r, you can see that the velocity of particle 1 will be proportional to gradient of 1 over r. That's basically like gravitation or electrostatic, but proportional to the activity of the second particle and the mobility of the first particle, and the velocity of the second particle will have the same functional dependence on the distance. But it will be proportional to the activity of particle number 1 and mobility of particle number 2. And unless you do something to make sure that the product of alpha 2 mu 1 is exactly the same as alpha 1 mu 2, and that is generically not going to be the case, then you will break action-reaction symmetry automatically in these systems. So that is a very powerful concept, because basically what I'm saying is as soon as you have this type of interaction, you have a non-reciprocal active system. And you can see that there are many consequences. There will be fluxes and dynamical currents that will persist in this system simply because of action-reaction symmetry breaking. Here you see a system that almost looks like an ionic liquid, so something which has valences that are basically in the positive, negative kind of category, and then they form what you can consider to be, ionic molecules like salt molecules, so sodium chloride and calcium chloride and so on. But they are self-propelled because they have this Janus structure once they form the complex, and also they are free to exchange monomers and so on. So you would get essentially molecules that form because of this non-reciprocal interaction, and at the same time, based on the symmetry that they acquire, they will exhibit some kind of non-equilibrium function like translational propulsion, spin or rotational propulsion, or other types of features. And here we can see the beginning of what looks like the paradigm that we have for proteins, because we know that the sequence that we have in the amino acid will determine the three-dimensional folded structure of the protein, which will then determine the unique function that they will have in the biological sense because of the structure that they could form. So here exactly is the same. By tuning the mu's and the alphas, you can determine what three-dimensional structure they will form, and then they will exhibit a very specific non-equilibrium function or behavior because of that structure. So here you can see an example where a slightly more complex molecule can exist in two different isomers, and it can do a barrier crossing jump between the two, and therefore you can very easily engineer with 3 catalytic colloids or 3 enzymes a system which propels itself and also exhibits run and tumble behavior, exactly like what E. coli does, without any sweating and waiting for a few billion years of evolution, basically. What about larger systems? So you can think about, in fact, you can think about the cytosol as being a very, very dense, a complex mixture of many components that are all catalytically, metabolically active, and they interact with each other. So they all have activities and mobilities, and they all create gradients around themselves when they are metabolically active. So what would be the natural structure that they would form? Would they stay stable or be unstable under certain conditions? So this is a very interesting question that we can start to explore. And of course, we can start simple. For example, here you can see stability of 2 species, depending on all the categories of interaction that you can find in this classification. I will start, so the axes are giving you mobilities and so on. But I will start by this one on the bottom right, explaining what the flavor of the interaction is. So in this parameter range, like particles repel each other and unlike particles attract each other. So this is almost like electrostatic interaction, even though the magnitude could be non-reciprocal. Up here, we have something that looks like gravity with positive and negative mass, because unlike particles repel each other and like particles attract each other. And then on this corner, we have 1 repels 1, 2 attracts 2, and 1 is chasing after, so is attracted to 2, and 2 is repelled by 1. So we see this chasing interaction, which creates frustration, for example. When we look at this binary mixture, already we can see quite exotic features. For example, this is a scenario in which a condensate is formed with a very specific stoichiometry between the 2 different types of catalytically active particles. And then when they form as a dense structure, they undergo a secondary instability, which would lead to the subsequent propulsion of the large structure. And then we can think about, for example, a possible role that they could play in self-organization of metabolic cycles themselves. So let's take citric acid cycle, what you see here, lots of enzymes interacting with each other basically in a cyclic manner. So every enzyme is catalyzing a reactant, which will then give rise to a product, which will be the reactant or the substrate to the next leg of the chemical reaction in the cycle. And then the question would be, can we say something about self-organization of metabolic cycles based on the basically on non-reciprocal interaction as a paradigm, whether it's possible for them to spontaneously form and exhibit this kind of cyclic behavior simply because of these non-reciprocal active systems. So we looked at this in detail, and in fact, it turns out that, well, you need to be patient. Just like any other thing, when you want to be serious about biology, you have to bring in, layers of detail. So we looked at cases where we have different chemicals, basically for the reactant and the product, for the substrate and the product, and different mobilities, and decided on the minimal model that would basically have all the elements. And this minimal model will end up having motifs like the ones you see here on the right, where particles might be in a chasing condition with the neighbors and self-attracting with their own species. And as soon as you have a structure like this, you start to have immediately very complex phase diagrams. One feature that comes out very interestingly as a consequence of this analysis is a sense of parity. Depending on whether the number of enzymes you have in the cycle is odd or even, you will have very different behavior. And this is something which, it's a prediction that you can also take to real data. I'll show you a video.
Time check, you're at about past 12 minutes.
Yes, so I have a couple of slides and I'll finish.
Sure.
Here you can see a situation with 5 enzymes where they basically come into a cyclic pattern, which have also this explosive behavior. It comes about simply because of the rules of frustration. You want to keep everyone happy, and this will create a cyclic behavior. So essentially, to summarize, the paradigm that can come out of this is that it's possible that we can explain the formation of metabolic cycles as being led by the information which would be already coded in the components that will basically live together in a metabolic cycle, and the active recruitment of them simply because of the non-reciprocity of the interactions. And this would be essentially similar to the central dogma of biology and the protein folding paradigm. So basically, what you use as the code of information will lead to naturally a very efficient way for the formation of that structure and the function that it will play later on. You can also think about other things like creating memory, self-organization by doing shape-shifting transitions between the memory, and then also studying field theories. Because I'm running out of time, I'll just basically skim through these last slides, and then maybe if there are questions, I can come back to them. Yeah, this one is my last slide. So essentially, the point of view that I would like to propose is that if we study functional modules about which we understand everything, and then we start building up the collective structures that will come from these modules interacting with each other, we can make predictions that could be tested in experiment. We could basically capture the essential low-dimensional subspaces where we need to focus and not be lost in the large space of parameters. And along the way, we can learn about new physics when we try to understand living systems from a physics perspective, and make scenarios and proposals for making living matter from the bottom up. So thanks very much. I think I can stop sharing.
Thank you very much. That was a great introduction to non-reciprocal interaction. Thank you. So we'll now move on to our third speaker, Guillaume Salbro.
So I have an issue with the screen sharing.
curious if it's working now for you, Guillaume.
I'm sorry, when I ask share screen, I don't get anything, unfortunately. Okay.
Oh, great.
Are you seeing my presentation?
Yes.
Okay, great.
Yes.
Sorry about the delay. Okay, so thank you very much for organizing this webinar, and thanks to Lisa and Ramin for sharing their insights on active matter in biology. And so on my side, I'm a theorist interested in connecting active matter physics to biology, and more particularly developmental biology. And so what I'm showing here are a few movies of developmental system. They are very colorful and dynamic. They show dramatic flows of cells which collectively move, decide for their fate, and establish the patterns and shape which are necessary to make up the structure of an organism, or in the case of the lower movie, to regenerate, lower left movie, regenerate a piece of the organism, and the bottom right movie is an organoid structure growing in vitro. And so because these structures, they are flowing and they are self-organizing, there is a connection to active matter that we want to use, and this will follow on some ideas that have already been put forward. But what's the link between active matter physics and biology? One idea is the emergence of collective behavior from many components, and there are many molecules in the cell and many cells in the tissue. Another idea is the system is active because it's out of equilibrium at the molecular level. So it's possible in principle to make such system artificially, for instance, by irradiating Janus particle with a laser. But also in a cell, there is activity, for instance, because of ATP hydrolysis, which provides the system with energy. And another connection is with soft matter physics is in liquid crystal, for instance, there are broken symmetry variables. There are molecules with a preferred direction. And in biological system, there are cytoskeletal filaments with a preferred direction and polarity, cell elongation. There are a lot of anisotropic structure that should be taken into account. And so how to describe these elements? So here I'm talking about something which is very familiar to physicists, which is hydrodynamics, and the idea to describe a system at the intermediate mesoscopic scale. And the classic example is water, which can be seen, water flowing in a pipe. That's a macroscopic question. The water is made of molecules at the microscopic level. And it turns out that a powerful way to analyze, to understand the physics of this system is to go at the intermediate scale and describe the motion of elements of fluids which contain many macroscopic particles, but which are small compared to the macroscopic system. And in the same way, in biology, we have motor, filaments, cross-linker, which are individual molecules, for instance, which make up the cytoskeleton of the cell. We also have many cells that make up an epithelium, which is deforming and flowing during development, like I was showing in my movie. And so the idea, one approach, one strategy to understand this link is to use continuum theories of active matter. So to put hydrodynamics of active matter to connect these scales. And one advantage in doing this is that then one identifies key variables, coarse-grained variables, which are important once we think about hydrodynamics. And these variables, they are, for instance, the velocity field, which tell us about flows and deformation. There are nematic tensor or polar vector fields, which tells us about anisotropic structures in the system. And there is the stress tensor, which tells us about mechanical forces in the continuum material. And then what one can do is one can write down constitutive equations for the stress, or for nematodynamics, in that case, for the evolution of nematic order. And these constitutive equations, they are phenomenological, so the terms are postulated based on the symmetry of the system. And they involve a number of coupling coefficients, which are, again, postulated. And the idea is then to measure them and to see some of them describe passive effects, some of them describe active effects. And the idea is to see how the control and pattern of these terms can allow us to understand how biological systems self-organize and develop. So this is one example. So in the left, this is a segmented movie of the gastrulation of the sea urchin embryo. In the right, it's an active surface simulation where here we have postulated a pattern of isotropic and anisotropic active stresses. And by comparing with the experiment, we have looked for such a pattern that would then explain the flow and deformation that we observed in the left. And in that way, we make a connection between active stresses and the three-dimensional morphogenesis that is observed here. Another very interesting variation on this is to ask about regulative active matter, so active matter with feedback. So these coupling coefficients that were visible in the constitutive equation I was writing down before, they can be internally controlled. So you can have active effects which will give rise to flows and deformation, and in turn, these flows and deformation, they can feedback on active effect because of concentration field that evolve with the flow or because of mechanical stresses that are sensed by the system. And this gives rise to a very rich set of behavior. This is just an example of a simulation of a growing tissue where the cell cycle is described using a Kuramoto model where the cell is coupled to its nearest neighbor. And because it's and the cells are growing, and because the neighbors are changing as the tissue is growing, there is such internal feedback that is present in the system. But I think this is a very large class of system, very interesting for biology to ask about how these internal feedbacks in active matter can explain self-organization. I was mentioning order parameter, and in the last years there have been many interesting studies and ideas around nematic and polar order in tissues. This is a movie of endothelial tube. This is a system that we have worked on the theory side recently, and so this is a microvessel on chip. It's an in vitro system, and when the cells that are lining the tube, it turns out that they have actin filaments which organize themselves along the axis of the tube. So they have nematic order, which one can quantify. And when pressure is applied to the tube, the filaments are rotating, and then they become oriented along the circumference. And in that way, they contribute to the stresses that oppose the application of pressure. So this is just an example of the importance of nematic and order that is present in biological system. But this is much more general, and another very rich idea is to look at living topological defect. So if there is order, there can also be defect, points at which the order is not defined, and there is a particular structure of the orientation field around the defect. And there are some remarkable observations, not from my group. In particular, the image on the right shows Hydra, an animal which has the capability of regenerating, and the body of the Hydra is lined by actin fibers which have topological defects at specific location on the surface of the animal, which illustrates the idea that the topological defect may play a role in orchestrating morphogenesis. When thinking about challenges, I thought about two things. One is, of course, to compare the theory to experiment, we need experimental measurements, which can be challenging. And one key information is to look at mechanical forces. And in the field of mechanobiology, many techniques, wonderful techniques have been developed based on traction form microscopy, chemical probes, droplet sensors, and in the left, this is an experiment with micropipette aspiration to apply a pressure to an embryo. But it's still quite an investment to be able to learn something about mechanical forces, and so that's something where technical development could be useful. Another question is, if we think about these continuum equations of active matter and we want to compare them to biological system, we often have to describe them in specific geometries, and that requires then numerical to solve numerically the equation, which can be challenging. This is these two the schematic on the left and the movie. They show finite element simulation of active surfaces that we have done to describe the interaction of a few cells. But I think there's also more progress to be made in that direction to have accessible numerical techniques to solve these equations. Perhaps a more general question that I find interesting is living matter a special form of active matter? So if we think that living matter is active matter, it is a subpart of active matter because it's we can make synthetic systems which are not living and are still active. And then what is the area of active matter that is occupied by living matter? And probably there are different ways of asking this question. For instance, it's relatively clear that living matter has capabilities of information processing that are not the case for any synthetic non-living active matter system. But another way to approach this is to ask, can evolution tell us something about which parameter space of active matter is living matter occupying? So can we explore the how evolution has played with active matter systems? And this is two slides from some work we have done with the ICME lab at EMBL, where we have looked at the shape diversity of a set of animals, of nidarians. So one can see on the left how the shape changes of these nidarians. And then we have compared this shape to active surface simulation description, where we describe the stress, internal stresses, and torques which are acting inside the tissues of these animals to find out the parameters that the active theory parameters that could explain the shapes that we observe. And doing this, we sort of build a connection between mechanotype and phenotype. So on the left, the mechanotype is the quantification of these active surface parameters and how they vary for different species. And on the right, it's the phenotype, so the actual shape, which is here quantified as a morphospace. And then by doing this, we can start to ask, if we compare different species, how are the active parameters changing? And this is perhaps another direction in which to go to do some evo-devo of active matter systems in biology. And just two other directions which I find are potentially interesting. Also, I'm not directly working on them. One is about energy consumption and fluxes and metabolism. So active matter requires energy. So how is energy consumption organized at the cellular tissue level? And are fluxes of energy important for cell and tissue self-organization? Are they constraining cell and tissue self-organization? And another interesting possible exploration is in bioelectricity. So on top of being active and nematic materials, can we also see biological tissues as active electromechanical elements? And is that important in some situations? Okay, and I would like to thank my group, which have been working on some of these questions over the years, and thank you for your attention.
Thank you very much. Thanks all the panelists for their excellent presentations. Now we'll move on to the panel discussion and Q&A section, and Deborah will kick us off there.
Thanks, Shreya, and thanks from me as well to all of the panels for those really insightful talks. I'd like to just remind the audience members that we encourage you all to submit questions using the Q&A box on the right of your screen. But before we get to that, we wanted to explore some broader issues with the panelists. So I'll kick off with the first question. This will go to each member of the panel. Perhaps, Lisa, you can answer first. So what, in your view, is the major bottleneck in terms of studies of active matter in biology currently? Do you view it as experimental or theoretical, or maybe as simple as like behavioural in terms of attitudes of researchers from across the two disciplines? Or is it something that even just depends on the particular context that's being studied?
Yeah, so I think this is an interesting question that may be context dependent. But the thing that one of the things that I see is that I do think that we've made really good progress on the statistical physics side, both in terms of, highly there's still many questions, but out of equilibrium systems and non-reciprocal interactions. I feel like on the theory side, we've made a lot of progress. But I think one of the areas where theoretically we don't really have a good handle on it is sort of a condensed matter or a solid state like physics, when you have strong interactions between the objects, and you really have the big difference with standard things is instead of having like two objects that always have a fixed interaction, or maybe even a fixed non-reciprocal interaction, what if you have a tunable interaction, sort of like Yelm was alluding to and that each individual cell has a tunable interaction with each of its neighbors that it can individually control. How do you think about the physics of, and it actually, and in my talk I alluded to it, the place where we've actually thought about that a little bit is, I think, carefully, is in machine learning and deep neural networks and in neuron systems, like how hot field type of models. So I think there's this sort of place for a lot of theoretical development in the space of thinking about a physics of objects where the interactions are really individually tunable. And I'm excited about that. And maybe I'll leave it there. There's other experimental and other things, but I'll just say that's one thing I think is interesting.
Excellent. And Ramin, over to you.
Yeah, so I think short answer is all the things that you mentioned are quite important aspects. You can actually ask the same question just about the field of active matter in itself, because there is a gap between the development from the theory side and from the experimental side. And then you can, of course, extend the question to biology, and that would bring in another community, which is the community of biologists, and the question of how to interface between these two. I would say the historical background is that active matter was a field that maybe started from the theory side, or at least it got into its growth or serious growth mode from the theory side. And it was a bit later on in the day that people started thinking about, okay, how do we actually test these theoretical ideas? And one of the challenges about non-equilibrium system is very, I mean, I can maybe put it this way. There are many more ways to be non-equilibrium than there are to be equilibrium, because equilibrium is just equilibrium, right? You're sitting in the happiest, memoryless configuration and don't care about how you got there. You don't care about more than 2 or 3 parameters as a system. So the system doesn't care about those things. But when you start to look at the same system, but in non-equilibrium conditions, then it depends how it got there. It depends even whether the conditions change rapidly or slowly, even that makes a difference. And then when you inject energy, it depends on how you drive it away from equilibrium. So all of these things mean that you will have very, very different types of non-equilibrium system, let's say, if you want to classify them. And that naturally means, people get lost, and essentially then it's very difficult to do a comprehensive study on that. And that's where I think connection to experiment could be very helpful, because then basically you would not get lost in the space of parameters. You would basically try to focus or let's say select the right small part of the configuration space where you can explore theoretical and experimental properties and ideas. And that's already, I mean, for that, I think the field of active matter has been catching up on that maybe in the last 10 or 15 years, but theory started even earlier than that. Then it comes with the biology question. I mean, that's a really big challenge, because again, short answer for that, I think, is it depends on the type of biology. There are some parts, for example, developmental biology, anything with mechanics. I think it's obvious to the biologists that the physicists are useful people to have around. But when it comes to some other types of biology, maybe it's not so obvious to them that they also need to work with physicists. And the quality of, let's say, cooperation between physicists and biologists will depend very strongly on, let's say, this need element, how seriously the physicists are needed for the particular question at hand. I think I'll stop at that.
Sure. And Guillaume, what are your thoughts?
Yeah, I mean, along the same line, I think active matter physics is very rich. We're seeing a lot of variations on it with interesting findings on active liquid crystals, active surfaces, control of active matter, non-reciprocal interaction, chiral active matter. There are many subfields which have amazing findings. And so if you ask to identify a bottleneck, maybe it's more on the experimental side to find ways to measure, especially in vivo, this the information that we need to compare to this array of possible theories. And maybe one particular, so that means improvement in imaging, measurement of mechanical forces, maybe other variables like electric fields. And I think also one thing is often in a biological system, one perturbs it, for instance, genetically, and then there is one or two perturbations. It's very difficult to have a continuous change in the control parameter that would allow to fully explore phase space. And sometimes in vitro it's possible. For instance, people confine tissues in chambers of different size, and that's a way to start to resolve continuously a control parameter. Or people build organoids in matrices with different elasticity, so that's also something that can be tuned. And I think this kind of experimental techniques that give a more fine way of controlling the system, they are exciting for further comparison between theory and experiment. And I also, yeah, I also agree with, I mean, myself, I'm mostly in contact with developmental biologists, and I think they see the interest of these approaches in their field, but perhaps in other fields of biology, it's not the same. I'm not sure.
I think we've, I was going to move on to another question that I think touches on some things that you've all been discussing there, but it's in studies of active matter in complex biological environments, how do you balance the incorporation of sufficient degrees of freedom to analyze the behavior in these complex contexts without over parameterizing? Raman, I'll start with you this time.
Yes. So, well, it's a great question. I think in the active matter community, people tend to talk about bottom-up approaches and top-down approaches. So, from the top-down view, in physics, it's customary to think about very broad constraints such as conservation laws and symmetries, and things that the systems will have to essentially follow, no matter what they are, no matter what details they will have. So conservation of energy, momentum, all of that, whether the matter is living or not, it doesn't matter. It has to follow all those things. So you start from those things and you try to constrain the space of possibilities to blocks where you would say in these blocks, the system will behave, let's say, more or less in a similar way. And then the bottom-up approach is to, let's say, start building up simple models. These are models that are stripped of many, many details, but they have, let's say, enough elements kept in them so that you can get the behavior that you're interested in, and essentially you can still have a manageable theory, and then coarse grain it, or let's say put it together in a way that can take you to the larger scale and can connect to those, let's say, symmetry properties and the more general properties that you have in mind. And in doing so, you would basically find a way to hunt for the right parts of the configuration space so that you don't have to worry about, many parameters and blindly, let's say, fit for them the way, let's say, in the machine learning community, people do things, and then they have to worry about overfitting and underfitting and so on. So traditionally, the approach has been like that, and even now that machine learning has entered this field, like any other field, still when people build, let's say, their models, they use these subspaces that are essentially guided by the symmetry principles and conservation laws, so that they don't have to waste a lot of time and energy searching places where they will be absolutely certain that they cannot, participate in the physical properties. So I would say physics-based approaches tend to, let's say, be more or less successful in this kind of thing. Of course, it depends also on the question that you're asking. So I know in biology, let's say if you want to design a drug for a certain disease, then of course details are the main topic, and then you will have to worry about how those details are integrated into these considerations.
Sure. Guillaume, did you have anything to add?
I think this was a very complete answer.
Yes.
And I think when I try to speak about hydrodynamics in my slide, hydrodynamic approaches, I think it's the same idea that using this approach, you limit the number of parameters and obtain description which are independent of many macroscopic details. And
Sure. And Lisa?
Yeah, in addition, I feel that it's been well answered, but a couple of things that I think are interesting questions are, I started to wonder, alluding to Guillaume's talk about evo-devo, if you have really important properties, for example, emergent properties for a particular developmental process, if those emergent properties were really dependent on the details of interactions between individual cells, not only would it not be robust, but also presumably it would be difficult to find evolutionarily. So I've sort of started to wonder if there are, there's a principle of universality that's useful in framing problems simply because the biology needs to find those solutions and maintain those solutions. In other words, if there's multiple ways to generate an emergent property and some emerge universally from features and others are dependent on details, the universal ones might be, end up being the ones that are found biologically. And then the second thing I wanted to mention is that one of the most fascinating results out of the AI literature that I think exists is that, people have realized that over-parameterization is not a problem in neural networks. It actually improves solutions.
Okay.
And so the other interesting thing that I've started to wonder is, are there, because Ramin, you alluded to the fact that in systems biology, you often have this problem where you overfit your data. But there are certain ways of framing problems where over-parameterization actually helps you a lot and regularizes the solution rather than causes the problem. And I wonder if biology doesn't set up the problems that way, so that the over-parameterization that each cell can fix its tunable properties individually becomes not an overfitting problem, but actually a regularization solution.
Great. Okay, over to Shreya.
Great. So I have a question, a little bit related to what was just said a little bit before, right? That this field started a little bit with theorists, using principles from statistical physics theory, for example, showing computational demonstrations in the field of active matter. So I'm curious to, get your thoughts a little bit on what are some challenges going forward for developing experimental platforms that realize and test theoretical predictions, and also some, interesting examples if you have in mind for where, experimental observations have confirmed or even refuted some theoretical predictions, for example. So maybe I'll start with Guillaume in this case.
Yes. So I think there is a difference between looking at biological systems, living systems, and in vitro reconstituted systems. I think for in vivo system, the difficult or the challenge or the excitement is in the making contact between what biological systems are doing and the theory. So because active matter physics is progressing, the theories are progressing, phenomena are being found, but in biology there are also deep questions which are unsolved. For instance, growth control, how the organ choose their size. There's a lot of progress, but not a full understanding, I would say. Or something that is fascinating is regeneration, the fact that some organisms are capable, when being cut, of reconstituting the piece of the body or the entire body. And the example of Hydra, which is an organism where you cut a piece, it regrows, and the fact that it has been found that nematic topological defects may be important for the regeneration process. That's a great example of where active matter physics and a deep question in biology can come into contact. I guess this requires a discussion between physicists and biologists. Now, when it comes about experimental platforms, then I guess it's more in vitro. And one thing is perhaps thinking about assembling collective of cells is going into, people have looked a lot into two-dimensional topological defect, and maybe I think that could be interesting going 3D, so looking at cellular assemblies in 3D and topological defects in 3D. Yes. And so you were asking about experimental realization that have confirmed theoretical prediction, and one story that came to my mind is so in 2005 there was a paper by Voituriez and co-worker where they showed that if you confine an active fluid in a stripe and you increase the size of the stripe, there is a transition at which the fluid will start to flow. And before, if the stripe is too small, it doesn't flow. If it's large enough, it starts to flow. And then in 2018, 13 years later, there was an experiment in the Silber's lab that showed that this was happening with using a stripe of cells. And what was interesting here is that is the time delay between the prediction and the observation, and also the fact that in the end it was done with cells, a particularly very complex object, not a reconstituted system where you think you would control every parameter.
Thank you. That's a great example. Lisa, I'll move on to you.
Yeah, just a couple of things, like what, as far as experimental platforms that I'd be really interested in. One of the things, Guillaume already mentioned that force measurements are really hard in vivo and would be so useful to constrain theories. And one of the places especially I see the need is we have all these sort of like fret-based sensors and sensors at the molecular level to understand molecular forces. But then there's usually a very big jump to sort of scales like ferrofluidic droplets or laser ablation. And it would be really exciting to have some more validation of how sort of, tension sensors that maybe even are going across an interface lead to like an interfacial tension at the scale of a cell or something that is, has some other properties. Like what's the relationship between those two types of force measurements at different scales? I think that would be really exciting. And then the other thing I would say, you already alluded to it too, Guillaume, is that I think development of careful biomimetic systems that could answer specific questions, and I'm thinking of like motion droplets coded in adherence, and those types of systems can be very helpful for teasing apart some of the non-biological sort of emergent properties that come out from activity, but not yet feedback, for example. And I think sort of putting together biomimetic systems right next to their biological counterparts and really teasing apart the differences is a nice experimental platform that could be developed further.
Thank you. Also, just for future, Lisa, we're losing audio on you a little bit, so but I'll move on to Raman for now.
Okay.
Yeah, so good answers so far. I mean, I would just add one thing maybe about in vitro experiments or experiments that need to essentially start from scratch, maybe using extracts of cells or completely synthetic material. I would go back to what Lisa mentioned in one of her slides. Dead matter is already hard enough, and imagine going this extra level and making it animated and then asking all kinds of questions about the behavior of these non-equilibrium systems. So already the constituents are very complex and delicate. For example, everyone who has worked with enzymes knows that they are very subtle, so they're very temperamental. You put them on a solid substrate, they stop doing what they normally do. So if you immobilize them, and if they are in the solution, they cannot be in a very crowded environment and so on and function in the same way. The temperature and the conditions and the pH and whatnot that they work on has to be very subtly tuned. If you mix them up, the conditions will not be happy for all of them simultaneously. Then in terms of microscopy, many of these systems involve chemical reactions that produce material like oxygen is one of them, and then they start forming bubbles, and then you start not seeing anything in the microscope very quickly. So you have essentially a fuzzy environment which is constantly moving around and regenerating. If you close it up, you would get a transient behavior, which will basically happen for a finite time and then die. If you create a stationary state, you will have to have inlets and outlets to pump in energy, and then that will bring in additional possibilities for leakage and all kinds of things. So these experiments, I mean, I would say on the scale of, history of science, active matter experiment has even not started yet. There are a handful of people doing really heroic things within conditions and the abilities that they have. Very disparate attempts. So I think for active matter to be successful as an experimental field, there has to be orders of magnitude improvement in the number of labs that join in and in the coordination of the efforts. Something like we see in solid state physics, in magnetism, in high energy physics, people doing national labs, doing big labs where people pass on knowledge and so on. So right now, a couple of you can call them, a couple of hippies doing things in their garages and so on. I mean, it's really at that stage in terms of how, and already they have shown us that there is a huge amount of potential and possibility. So I would say this field hasn't even started.
Thank you. We have quite a few audience questions. So Deborah, I'll hand off to you to bring some of that to the panelists.
Okay, sure. So we have one from Rudy Winkelbauer, and he asks, with so many parameters involved, how many identical looking solutions are possible? Could this undermine the active matter approach in biology? So Lisa,
I think this is such a fantastic question, because I've been thinking a lot about that exact question. So one of the interesting things actually is it might be the opposite, in my opinion, that in the same way that, and I know I've been using this analogy a lot, but I've been thinking about it a lot. In the same way that if you have an over-parameterized machine learning deep neural network, you still get a really good solution to the problem, even if you reinitialize all of the weights. The solution has a different set of weights when you solve the problem in the training, and yet you still get a really good answer. And so rather than thinking about that as a flaw, I think about it as possibly the solution, in the sense that what it teaches us is just like a glassy potential energy landscape in glasses. There are a lot of good, kind of equivalent metastable states that are good enough. And that's kind of nice. And the other, so in some sense, what it means is what the value of each individual parameter is not so important. It's just the statistics of the collective needs to be appropriate to get you into one of those good enough solutions. And so actually there's a lot of hope, in my opinion, that there are these equally good solutions are an ensemble that we should understand the properties of.
Sure. Did anyone else want to chip in with thoughts there?
Yeah, I can just add to that in history also we've had examples where people showed that when things get really too complicated, maybe we can make progress by just assuming that they are random. We've had this in nuclear physics where people just use random matrix theory to make very precise predictions about structure of nuclei. And then the same kind of theory was used by Robert May to basically set up these concepts for evolution and stability of populations that are basically interacting with each other in such a complex way that if you can just assume they are random, and then already you can make very good progress. So in fact, we have used the same approach for non-reciprocal active matter with multi-component system using random matrix theory, and you do get similar kind of progress. Plus, when you have this notion of, let's say, dependence on initial condition, it's often the case that when you have noise basically washes out over all these sensitivities of dependence on initial conditions. So somehow maybe the complexities will look after themselves and create smaller sort of number of possibilities, let's say.
Yeah, and so,
Yeah.
yeah, so to this, what this makes me think of is so there's this sort of mechanical degeneracy that if you observe a shape, you can have many different state of stress that will explain this shape, and that makes inverse problem of finding the stress that explain the shape difficult. So one thing is you can measure the stress, and then you know what state of stress you actually have. Another thing is, I think that links to what Lisa was saying earlier. It's also interesting in itself. Is biology playing with this degeneracy to have different solutions to the same problem? For instance, different state of stress giving rise to the same shape. Is this actually happening?
Okay. Back to you then, Shreya.
Sure. I'll ask a question from the audience member, Yashumati Ratan. Can we manipulate active matter? And maybe, if you can give us some short examples of, some examples of that. I'll start with Ramin in this case.
Yes, in fact, controlling active matter has been a very active field, both in theory and experiment. So there are many people who use optical traps to create, for example, you can make holographic patterns of the intensity of the optical landscape. You can even change that dynamically, and then both in synthetic systems where you would use optical, so accumulation of light to trap particles and manipulate them. You can also use optogenetics to basically address individually. So there are now, examples where experiments where bacterial suspensions are optogenetically addressed to form different shapes, and collectives of bacteria as a response to the light intensity pattern. I don't know. You can make Einstein face that conforms into Darwin and stuff like that, actually using this kind of manipulation. So the answer is yes. It's a very interesting development.
Guillaume or Lisa, if you have something to add here, you're welcome to.
Yeah, I think autogenetics is a very, talking about cells and tissues, I think autogenetics is a very promising strategy because it can be applied in vitro, but also in vivo to perturb in a controlled way the system. And I think there is some higher promise in that technique.
Yeah, it's almost as if biology systems are better for this because they are able to be, you can manipulate it with optogenetics pretty well, whereas the active matter, physics systems have to catch up in that controllability. On that end, though, I wanted to highlight some, like Mark Miskin has these amazing micro robots that can interact with one another and are really highly programmable and individually addressable. And they're like, you make that, you fabricate them on with using computer chip technology. So they're really tiny. And then there's also manipulation of magnetic particles by magnetic fields in active matter systems, in addition to the optogenetics. So I think on the active matter, physics material side, there are some promising, highly controllable active matter platforms, but I would say they're not yet at the point of optogenetically controlled cells.
Deborah, maybe there's time for one more short audience question.
Sure, I'm trying to find a general one. So Armin Tajik has asked, in 50 years, what's the single biological active matter question you'd like to see an answer to? Perhaps we can have some just little short answers to that one. Maybe it's too thought-provoking to give a short, quick answer to.
I mean, I could jump in really quickly. I think this is, I don't know whether it's what the question asked, but I think it's really interesting to think about understanding enough about how biology does morphodynamics to really make shape-shifting bio-inspired materials that are closed loop. So I think there's this really big question of what are the minimal ingredients you need to have kind of not just functional shape changes, but sort of dynamic materials that are sort of like a robot material hybrid. And I think we would learn a lot from biology about how to do that.
Yeah, just the thing that comes on top of my head is I was talking about regeneration. I would like to see how regeneration can be understood as an active matter system.
Sure. Ramin, anything to add?
Yeah, I think so. I don't know about what would be possible in 50 years, but I think for me the best question to ask from active matter side is, why is life possible at all? I mean, we can of course debate about what happened at the early stages of the origins of life and so on, and there will be very little evidence to actually corroborate any kind of theory that one might have. But at the end of the day, we can draw a lot of, we can build constraints and conditions that will shape the space of possibilities. And then if that space of possibilities becomes, finite, then we have answered the question, why can life exist at all from the physics point of view? And I think that would be good enough.
Thank you, everyone. I think we are past the time, so I believe we've come to the end of our session. Thank you so much for all of our speakers for engaging presentations, their panel discussion. Thank you, all of you in the audience, for tuning in today to learn more about active matter in biology. Thank you so much for your engaging questions. If you missed anything during this webinar or would like to listen to it again, a recording will be available shortly, and it will be posted on cell.com. Please take a few moments after this webinar to complete a brief survey that will be emailed to you momentarily. Your comments will help us improve our webinars, and we really value your feedback. Thank you again to everyone, and we hope to see you all at a future CellPress event.
Thank you.
Thank you. Bye-bye.